Moving on to 21.4, we are going to look at penicillin and how it is created in a fermenter or a bioreactor. Penicillin is an antibiotic. Many of you have taken antibiotics throughout your life to treat various types of infections. Antibiotics are a substance that kills bacteria without harming human cells. What antibiotics actually do is they attack cell walls as opposed to cell membranes. So, something that's unique about a bacteria cell has a cell wall. Human cells do not have cell walls. Think back to our unit on the cell in itself. Human cells just have that cell membrane. Bacteria cells are similar to plant cells in that they have the cell wall. So, what an antibiotic does is it attacks the cell wall. If the bacteria cell does not have the cell wall, then it ultimately will be destroyed. So we take antibiotics to help cure any type of bacteria infection. The most common type of antibiotic is something called penicillin, which is made from growing the fungus Penicillium. So if you think back to our first video on biotechnology, we can harness using fungi and different types of fungus to ultimately create things that are very helpful for us. One being antibiotics, which is probably one of the greatest scientific discoveries known to man. Just being able to treat basic infections that at one point in history would have allowed before they were created, so many people died from infections that were really just basic, but now we're able to just take a round of antibiotics and be on our merry way. Now, when we look at penicillin, this is how it is created. It's created in the fermentation process by using a fermenter, which is basically a device that once we have a food source, which remember, sugar needs to be added because it is being created from a fungus, so think like a yeast. And we're going to have those carbohydrates. Amino acids are going to also be placed in there for a source of nitrates. There will also be provided oxygen, as well as steam or some sort of water to control temperature, which is very, very important. So the pH, the temperature, all of these outside factors are controlled to ultimately have these the fungus of Penicillium mixed around in this fermenter, and then the final product, once everything is complete, is a culture of penicillin that is extracted at the bottom of that actual device, the fermenter. Okay, now this is a process that takes about 15 to 24 hours, so it is something that takes a little bit of time. Everything has to be controlled. The amount of sugar has to be controlled. The amount or the rate of production can slow down if all these conditions are not optimum. So ultimately, this is something happening in a lab that is very controlled and harnessed to ultimately provide for us life-saving antibiotics.